Hi, my name is Angham Ahmed. I'm a biology master's student at California State University, Fresno, working in Dr. Corey Burke's lab. First of all, I would like to thank you for allowing me to be part of this amazing conference and meeting great-minded scientists. Today, I'll be talking about my research project, and the topic is distinctive binding mechanism of Camelid VHHs to Intimin of Enterohemorrhagic E. coli O157H7. Enterohemorrhagic E. coli is a food-borne pathogen. Cattle is their main reservoir but can be transmitted to humans through the consumption of contaminated food product. EHEC pathogenesis of the human intestinal tract involve two critical virulence factors called Intimin and Ter, and their interaction is very critical because it results in a tight binding of the bacteria onto the host cell membrane. This is a zoom-in picture of the Intimin and Ter interacting, and currently there is no treatment against this pathogen. One possible solution would be using single-domain antibodies, which are the antigen binding domain of heavy-chain only antibodies naturally produced by Camelid family. Intimin-specific VHHs that were generated through the immunization of a llama, construction of a phage display, and selection for positive clones, showed high specificity and binding affinity for Intimin. Therefore, we hypothesize that these nanobodies would have the capacity to neutralize EHEC pathogenesis. And our research goal was to determine the complex crystal structure of these VHHs bound to Intimin to unravel their binding mechanism using an approach called X-ray crystallography. Collaboration with Dr. Henry at the National Research Council of Canada, SPR epitope binding experiment were carried out to see if all of our 5 VHHs bind on the same or distinct epitope of Intimin. The table here shows the number of cycles, also indicated by the red and green colors, and the number of injections. The antigen was covalently coupled to the sensor surface of the machine, and VHH2 was injected first at time 0, and then we see a response. After 150 seconds, a mixture of VHH2 and VHH5 was injected, and we see a second response. Cycle number 2, VHH5 was injected, and we see a response. Again, after 150 seconds, for the second injection, we see a second response of the mixture VHH2 and VHH5, which indicate that these two VHHs bind on a distinct epitopes on Intimin. And this sensogram, we see here that for the first injection, we see a strong response for both cycles 3 and 4, but in injection 2, we see a small response, which indicate that these VHHs bind on an overlapping epitope. Summary of the experiment is that VHHs or 3 VHHs bind on the same epitope, and VHH2 is overlapping with these VHHs, while VHH5 binds on a totally different epitope. Here, we determine the complex crystal structure of 3 VHHs bound onto Intimin. The VHHs are color-coded and are superimposed, as can be seen here. Structural data showed that these VHHs form polar contact with Intimin using CDR3 and framework 2 paratopes. The structural data also revealed or confirmed previous findings of SPR epitope binding, that these VHHs bind on the same or at a overlapping epitope on Intimin. The major finding or the structural data also revealed that these VHHs probably lack the neutralization capacity of EHEC pathogenesis. As can be seen here, the VHHs bind on an adjacent epitope of Intimin, not on the Ter binding domain of Intimin, blocking their interaction. So this refutes our hypothesis that these VHHs have the capacity to neutralize EHEC pathogenesis. Another major finding of our research project is that our VHHs exhibit a distinctive binding mechanism. They bind to Intimin in a sideways orientation, as can be seen here, with framework 2 region protruding towards the antigen, while CDR3 loop is facing away. The involvement of CDR2 loop is probably to compensate for the lack of binding of the other CDR loops. Framework 2 region is the former VL interface of VH of classical antibodies. In VHHs, the residues here are mostly hydrophilic and are probably increasing the solubility of these VHHs. Specifically, arginine is one of the canonical VHHs solubility residues. So maybe this framework region has been shaped this way by evolution to increase the solubility, also to be able to form a polar contact with antigens.